Welcome back to the deep dive. Today we are undertaking a really foundational journey. We are zooming past the simple idea of cells and organelles and diving, way down to the molecular level.
hmm
Our goal is to uncover the pure chemistry that provides the bedrock of all biology.
Hmm.
And this isn't just, academic groundwork.
Not at all.
We're examining the molecular architecture, the actual physics and chemistry that allows life's machinery,
Mhm.
from your muscle contraction to cell division, to exist at all.
That's the mission, really. We are using Chapter 2 of CARP Cell and Molecular Biology as our source map today, and the goal is to provide you with the necessary chemical foundation to understand all the complex activities of the cell.
Right.
Because if we don't get how molecules interact, how their bonds are formed, and, why certain shapes are favored over others, then the rest of cellular biology just doesn't make sense.
And to really start this story, we have to go back in time. All the way back to Earth, 4.6 billion years ago. We're talking about the chemical origin of life itself.
You have to picture the early Earth. It was a chaotic, a truly tumultuous environment, violent volcanic eruptions, constant asteroid collisions. Just a mess.
And yet, life somehow got started.
It did, and remarkably quickly. We have evidence from ancient microbial formations that suggests life could have emerged as early as 3.5 billion years ago.
So that's the fundamental puzzle, isn't it? How do you bridge that gap? How do you get from a planet that's just inorganic chemicals and rock to the organized, self-replicating complexity of a cell?
Well, researchers hypothesized that the earliest structures, which they call protocells, were incredibly simple.
Okay, what does simple mean here?
Maybe just some genetic material, perhaps RNA, surrounded by a very primitive membrane. But even before you can assemble that, you need the building blocks.
The parts list.
Exactly. The challenge was proving that these organic components, amino acids, sugars, could actually form spontaneously under those early Earth conditions. And this leads us to the landmark 1952 Miller-Urey experiment.
Oh, the famous lag simulation, the one that mimicked the presumed early atmosphere.
That's the one. Stanley Miller and Harold Urey constructed the sealed glass apparatus. Inside, they put water, methane, ammonia, and hydrogen. All the gases they believed were abundant back then.
And then they added a spark.
They did. They introduced energy in the form of heat and electrical discharges to simulate, lightning and volcanic activity. The discovery that followed was genuinely profound.
It's a spontaneous creation of biological molecules.
After just 2 weeks, the apparatus was coated with these complex organic compounds. And crucially, they had spontaneously generated fundamental building blocks like amino acids, which make up proteins, and various sugars.
So this was huge. It strongly supported the idea that the conditions on the young planet were, well, they were ideal, maybe even mandatory, for generating the organic precursors of life.
Absolutely. And modern research has expanded on this. We now have plausible prebiotic pathways for other essentials, like ribonucleotides for RNA and even the fatty acids you'd need for membranes.
So if we can create the building blocks in a lab simulation, we must be getting close to assembling a protocell. But the source highlights a deep chemical mystery that still persists, the issue of chirality.
Oh, this is a major, major structural challenge for any origin of life theory. When the Miller-Urey experiment produced amino acids and sugars, it produced them as an even mixture of left and right-handed isomers.
Mirror images.
Exactly. Molecules that are mirror images of each other. The classic analogy is your left and right hands. They are identical, but you can't superimpose them.
Okay.
Yet all known life on Earth is incredibly selective. It exclusively uses left-handed amino acids and right-handed sugars.
So why the bias? How does that chemical selectivity happen? Did a meteor impact or some other early event introduce a preference?
That is the big open question. We just don't know the initial chemical trigger that caused this fundamental selectivity. But the fact that every single living thing shares this preference for one mirror image, one chirality, it strongly suggests that this selection happened once, very early on, and it became fundamental to all subsequent biological processes.
All right, so let's move from cosmic history to the subatomic forces that are required for life to even endure. Everything we've discussed, from those protocells to DNA, it all depends on incredibly strong, stable connections. We're talking about covalent bonds.
Covalent bonds really are the stable foundation. They form when atoms share pairs of electrons to fill their outermost electron shells, which is how they achieve stability.
The water molecule is the classic example, right?
It's the perfect example. Oxygen has 6 outer electrons, and it satisfies its need for 8 by sharing electrons with two separate hydrogen atoms.
But here's a question. If thermal energy is constantly jiggling molecules around inside the cell, how is it possible that complex structures like DNA or proteins stay intact long enough to be useful?
That's where the strength comes in. And this relates directly back to the physical requirements for molecular survival on that early, tumultuous Earth.
Okay.
The energy required to break a typical single bond, say a carbon-hydrogen or a carbon-carbon bond, is enormous. We're talking 80 to 100 kilocalories per mole.
And how does that compare to the thermal energy?
Well, now you compare that to the random thermal energy that's being exerted on a molecule in the cell, which is only about 0.6 kilopon.
Wow. Okay, so it's not even close.
Not even in the same ballpark. This vast difference means that random thermal vibrations are simply insufficient to break those covalent bonds. They are stable, rigid anchors under biological conditions.
And the way these electrons are arranged in their orbitals, which, we can visualize from diagrams like figure 2.1 in the text, that's what dictates how different elements behave.
Absolutely. The number of electrons an atom has determines its bonding behavior. Take carbon and silicon, for instance.
They're the same column on the periodic table.
They are. Both have 4 outer shell electrons. Both can form 4 bonds. But because carbon is a much smaller atom, its nucleus pulls the shared electrons very strongly. This allows it to form the vast, stable, long-chain organic molecules that really define life.
And silicon can't do that.
Not comparably, it's just too large. And then you have the other extreme, elements like neon and argon. Their outer shells are completely filled, which is why they are inert gases. They have no drive whatsoever to form bonds.
So carbon forms this stable backbone, but the bonds aren't always single bonds. We see double and triple bonds, and these impose some serious constraints on a molecule's shape.
Right. Double bonds, like in oxygen gas, involve sharing 2 pairs of electrons. Triple bonds, like in nitrogen gas, share 3. And the crucial structural consequence of this is rigidity.
How so?
A single bond allows for free rotation of the linked atoms. They can spin around. Double and triple bonds prevent this rotation completely, locking the atoms into a fixed geometry.
And that rigidity is important.
Incredibly important. It's often where biological mechanisms capture and utilize energy. We'll see this later when we talk about photosynthesis, as the text hints at with a reference to figure 6.6. That rigidity is key.
Okay, so now, when we start linking different elements together, we introduce the concept of electronegativity.
Yes, and this is just the nucleus's attractive power, its pull, on those shared electrons. When 2 identical atoms bond, like 2 carbons, the sharing is perfectly equal.
But life isn't made of just carbon.
No, life uses highly electronegative atoms like nitrogen and especially oxygen. And what they do is they pull the shared electrons closer to themselves.
And that uneven sharing leads directly to polarity, which feels like it's fundamental to pretty much everything that happens in the cell.
It creates what's called a dipole. In a polar molecule like water, the highly electronegative oxygen atom acquires a partial negative charge because it's holding on to the electrons longer.
Which leaves the hydrogens with a partial positive charge.
Exactly. This asymmetric charge distribution is what makes molecules highly reactive. And conversely, molecules that are composed primarily of carbon and hydrogen, like the hydrocarbons you find in waxes and fats, are nonpolar. They lack these strong dipoles, and that makes them relatively inert.
But if that electronegativity difference is great enough, the atom doesn't just share the electrons unequally, it strips them away entirely. And that leads to ionization.
That's how we get true ions, which are stabilized by having their shells filled. For instance, sodium easily loses its single outer electron to chlorine, which readily accepts it.
And you get table salt.
You get sodium as a positively charged incals, +5, and chlorine as a negatively charged anion. They form stable salts. But not all single electrons are stabilized. Some lead to highly destructive species, free radicals.
This brings us to the human perspective box in the chapter. The free radical hypothesis of aging is, well, it's pretty pervasive. The idea is that aging is essentially the cumulative damage caused by these highly reactive chemical species attacking our cellular components, particularly our DNA.
And a free radical is, simply put, a molecule or an atom that has an orbital containing a single unpaired electron.
And it wants a partner.
It desperately wants a partner.
Yeah.
Because they're chemically driven to pair that electron, they become highly unstable and extremely reactive. They can form when a covalent bond breaks and each atom keeps one of the shared electrons, or during oxidation reduction reactions when an atom accepts just a single electron.
So how do they form inside us and what makes them so destructive?
Well, they form continually, especially in our mitochondria during normal oxidative metabolism. The book notes that about 1 to 2% of the oxygen we breathe ends up as hydrogen peroxide, text O2 or as the superoxide radical instead of water.
And their destructive power.
It comes from their reactivity. They chemically alter proteins, lipids, and nucleic acids. In fact, our own immune cells leverage this destructive power to kill ingested bacteria. And the hydroxyl radical is generated by sunlight. That's what's responsible for skin damage.
So the body must have evolved some pretty robust defenses to continuously disarm these things.
Oh, absolutely. The pivotal discovery here was made back in 1969 by Joe McCord and Erwin Friedrich. They identified an enzyme called superoxide dismutase, or TextOD-DO.
And what does TextSOD do?
TextOD's only job is to destroy the dangerous superoxide radical. TextOD catalyzes its conversion into oxygen and hydrogen peroxide.
Okay, but hydrogen peroxide itself is a strong oxidizing agent. It's the stuff we use to disinfect wounds, so the cell needs a secondary line of defense to get rid of that.
Correct. And it has one. The cell employs other enzymes like catalase or glutathione peroxidase to rapidly destroy the TextO2-DO. The importance of this whole defense system is demonstrated really dramatically in genetic studies.
How so?
Mice that are engineered to lack the mitochondrial form of TextSOD, which is called TextSOD2, die very, very quickly after birth.
And the most compelling experimental evidence supporting the aging hypothesis came from reversing this process, right?
Yes, this was a major breakthrough in 2005. Scientists engineered mice to overexpress the TextO2 destroying enzyme catalase, but specifically within their mitochondria.
And the result?
These mice lived 20% longer, on average, than their control litter mates. This was a clear demonstration that enhancing these antioxidant defenses could actually extend the lifespan of a mammal.
But the source is careful to note that the free radical hypothesis as the sole driver of aging is still debated. It's competing with this idea of programmed aging.
It's an ongoing scientific argument. Programmed aging suggests that an organism's decline is actively managed by evolved mechanisms, maybe to favor reproductive fitness over sheer longevity.
Which is different from just random accumulated damage.
Very different. And what's more, the clinical application, the massive industry surrounding antioxidant supplements like vitamin E, vitamin C, and glutathione, has largely failed to provide convincing evidence that these dietary supplements actually retard the aging process or increase maximum lifespan in mammals.
So the toxicity of the radicals is undeniable, but whether they are the master clock of aging.
That remains controversial.
Okay, so from the uncontrolled chaos of biological free radicals, let's pivot now to how scientists harness that exact same fundamental atomic instability, isotopes, and radioactivity, but for controlled medicine.
It's a fascinating application. So every atom of an element has a fixed number of protons.
Right. For imaging, for instance, radionuclides are used as tracers that you can inject into the body.
The most common example is probably technetium-99, or textjetchen. It's a gamma emitter, and it's commonly used for things like bone scans. It's really ideal for this because the energy of the gamma ray it emits is similar to a diagnostic X-ray, and it has a relatively short half-life.
Which minimizes the patient's exposure to radiation.
Exactly.
But that short half-life must make shipping it incredibly difficult. How do hospitals solve that logistical puzzle if it's decaying so quickly?
This is a fantastic example of biomedical engineering ingenuity. Instead of shipping the short-lived text itself, suppliers ship a generator containing a different isotope, molybdenum-99 or TEXMO.
And the molybdenum decays into the technetium.
Precisely. The TEXMO continually decays to produce TEXMO. So when the hospital needs a tracer, say, for a brain scan, using TEXtup that's been chemically modified to cross the blood-brain barrier, they simply extract the freshly made TEXtup from the generator.
That's very clever.
It is. And we use other tracers too, like indium and gallium.
Right.
These can be chemically linked to antibodies that are designed to specifically seek out and bind to tumor cells, essentially lighting them up for visualization.
And the other key medical application is, of course, therapy, using the destructive power of radiation to target and kill tumor cells.
Right. This includes external beam therapy, which often uses strong gamma emitters like cobalt-60, textcolite. A really sophisticated example of this is the gamma knife technique, which the text describes with figure 2.2.
Can you walk us through that?
Sure. Imagine a hemispherical array, almost like a helmet, with many individual beams of gamma radiation. All of these weak beams are focused with incredible precision onto a single point deep within the body, like a brain tumor.
So the dose is concentrated only at the target.
Exactly. This concentrates the destructive dose exactly where it's needed while sparing all the surrounding healthy tissue.
And there's also internal radiation therapy or Bragg therapy.
Yes, that involves implanting small, localized pellets such as iodine-125 or directly into the cancer's tissue. This is often used for prostate cancer.
But what's crucial to understand is how the destruction actually occurs. The gamma rays usually don't break the cancer DNA directly, do they?
No, it's a secondary mechanism, and it's actually very similar to the action of free radicals we were just talking about.
So what's happening at the molecular level?
When the gamma rays hit atoms in the tissue, they expel fast-moving secondary electrons. These electrons are the true agents of destruction. They either physically break the bonds in the DNA helix upon impact, or they ionize nearby water molecules in the cell, creating those highly destructive hydroxyl radicals that then chemically attack the DNA. It's a very precise, controlled application of fundamental molecular physics. Hashtag tag 2.3 noncovalent bonds.
We've established that covalent bonds provide the strong, stable structural framework of all biological molecules. But life is inherently dynamic. Molecules have to interact, change shape, and separate constantly. And this fluidity is governed by noncovalent bonds.
Exactly. Non-covalent bonds are the weaker dynamic linkages. Individually, they are quite fragile. We're talking a range of 1 to 5 kilocalories per mole.
So they're easily broken.
Very easily broken and reformed by the constant thermal energy in the cell. And that weakness is what allows for the high speed and dynamic interactions that define life.
But if they're so weak individually, how can they possibly hold together massive structures like DNA?
Because their effects are entirely additive. When large numbers of these weak bonds act in concert across the vast surface of a complex structure, like the tens of thousands of bonds holding the two strands of a DNA molecule together, which we see in figure 2.4, they provide enormous overall stability.
But they retain the ability to be broken when needed.
That's the key. Stability, but also reversibility.
Let's review the main types, starting with ionic bonds, or as they're sometimes called, salt bridges.
These are simple electrostatic attractions between fully charged ions. They are very strong in a vacuum or a dry crystal, like we see in figure 2.3.
But not in the cell.
Right. Inside the cell, which is mostly water, they are considerably weakened. Water molecules surround and insulate the charged ions, which prevents them from getting close enough to form really strong linkages.
But they're still important.
Oh, yes, they're still very important for large macromolecules. Think of the ionic bonds between the negatively charged phosphate backbone of DNA and some positively charged amino acid residues on a protein surface.
Okay, next up, the ubiquitous hydrogen bonds.
Hydrogen bonds. They form when a hydrogen atom that is already covalently bonded to a highly electronegative atom, usually oxygen or nitrogen, develops a partial positive charge.
And that partially positive hydrogen is then attracted to what?
It's attracted to the unshared pair of electrons on a neighboring electronegative atom. The text has a great illustration of this in figure 2.5. Individually, they are extremely weak, often just 1 kilomole, but they are absolutely crucial for holding complex molecular shapes and, of course, for giving water its unique properties.
Then we arrive at hydrophobic interactions, which, and we have to remember this, are not actual bonds of attraction.
This is a crucial distinction. Hydrophobic molecules, fats, steroids, they, for lack of a better word, hate water. When you place these nonpolar molecules in water, they spontaneously aggregate together.
But not because they're attracted to each other.
Not at all. It's because they are minimizing their contact with the surrounding polar water molecules. The water molecules, in turn, are maximizing their own disorder, or entropy, by excluding the nonpolar surfaces. Figure 2.6 shows this really well.
So it's pure exclusion driven by the water molecules trying to organize themselves.
Precisely. Just think of that next time you watch a fat droplet reforming on the surface of your soup. That's maximum entropy at work. The water is literally pushing the oil together. These interactions are fundamental for driving protein folding and, critically, for forming the basic structure of all cell membranes.
And finally, the weakest links of all, van der Waals forces.
These are fleeting, extremely weak attractions ranging from just 0.1 to 0.3 kcal. They're highly distance sensitive.
And where do they come from?
They arise from temporary transient asymmetries in electron distribution. This creates momentary dipoles even in nonpolar molecules. Though they're negligible on their own, they become really significant when two macromolecules have complementary shapes, allowing thousands of atoms to approach each other very closely.
Just so when all these weak forces accumulate.
They generate enough collective attraction to stabilize an interaction. The example in figure 2.7b is a large antibody binding tightly to the surface of a viral protein. It's the sum of all those tiny van der Waals forces that holds it there. Hashtag tags the life-supporting properties of water.
The environment where all these bonds operate is, of course, water. Text's unique chemical structure is what makes life as we know it possible.
Absolutely. Water's properties, its asymmetry, its highly polarized bonds, and most importantly, its capacity to form hydrogen bonds with up to 4 other water molecules, as you can see in figure 2.8. It all comes together to create this extensive, interconnected fluid matrix.
And this network is directly responsible for its unusual thermal behavior.
Yes. Because water molecules adhere to each other so strongly, a massive amount of energy is required just to break those hydrogen bonds before the temperature of the water can rise significantly.
And this is why sweating cools us down.
Exactly. This high heat requirement for evaporation is why sweating is such an effective cooling mechanism. The heat that's required to break the bonds and evaporate the liquid is absorbed directly from your body.
So water isn't just the universal solvent, it's actively dictating the structural dynamics of life.
It dictates everything. It dissolves polar substances, it maintains the separation of ions, and it forces nonpolar molecules to fold inward. It's also crucial for maintaining macromolecular shape. There's a great image in the text, figure 2.9, that shows ordered water molecules acting as internal structural components.
What's the example?
It's a clam hemoglobin molecule, and you can see these water molecules forming hydrogen bonds between different subunits of the large protein, helping to stabilize its precise functional configuration. It's an active participant. Hashtag 2.4 acids, bases, and buffers.
The viability of all this cellular activity depends critically on the chemical environment. Specifically, the concentration of protons, which is the basis of pH.
Right. We have to remember that the cellular environment is defined by its acidity. In that environment, acids are always looking to donate a hydrogen ion or a proton. Think of acetic acid dissociating in solution.
And conversely, a base accepts a proton.
Like in the text NH2 group in a protein. And these always exist as what we call conjugate pairs. An acid becomes its conjugate base after it loses a proton.
And some molecules, like water and amino acids, are amphoteric. They can act as both.
That's right. And acid strength is simply a measure of how easily that proton is lost. Hydrochloric acid is considered strong because it readily transfers its proton to water, whereas acetic acid is weak because it remains largely undissociated.
And we measure all of this on the pH scale. Because it's logarithmic, a small change on the scale is actually a huge shift in proton concentration.
Yes, pH is the negative logarithm of the hydrogen ion concentration. This means a shift of just 1 pH unit, say, from 7 to 6, reflects a 10-fold difference in the proton concentration.
Which is why biological processes are so sensitive to pH fluctuations.
Extremely sensitive. Too many protons, and they might, for instance, protonate a critical text NH2 group on an amino acid residue. That would instantly change a protein's charge and structure and potentially destroy its activity.
So the cell needs chemical shock absorbers to prevent these rapid damaging fluctuations. We call these buffers.
Buffers are always a mixture of a weak acid and its conjugate base. They work to stabilize pH by reacting with any excess protons or hydroxyl ions that appear.
The blood buffer system is a primary example.
It is. It's based on carbonic acid and bicarbonate ions, and it's what maintains the stable blood pH of about 7.4. If, for instance, exercise generates excess acid in your blood, bicarbonate ions immediately swoop in and remove those excess protons from the solution, keeping the pH constant.
And there's a different system inside the cells.
Yes. The intracellular fluid uses a similar but distinct phosphate buffer system to do the same job. Hashtag 2.5, the nature of biological molecules.
Okay, with the underlying chemistry established, bonds, water, buffers, we can now look at the 4 major classes of molecules that life is built from, starting with carbon's unique role.
The modern definition is pretty straightforward. Organic molecules contain carbon-carbon bonds. Biochemicals are those that are specifically produced by living things.
And carbon is central to all of this.
It is. With its 4 outer electrons, it can form 4 stable covalent bonds, linking to other carbon atoms in virtually limitless backbones. These can be linear chains, highly branched structures, or stable rings like the 4-ringed structure of cholesterol you can see in figure 2.10.
And as we noted earlier, no other element offers this kind of structural flexibility.
Nope, it's unique.
But that inert hydrocarbon backbone needs some specialization. Reactivity is conferred by what we call functional groups.
Yes, functional groups are specific groupings of atoms that replace a hydrogen, and they are what provide chemical reactivity, polarity, and water solubility. You can see a list of them in Table 2.2. They often contain the highly electronegative atoms, O, N, P, or S.
And they dramatically alter the properties of the molecule.
Dramatically. For example, if you take the simple hydrocarbon ethane and replace one hydrogen, you get ethanol, which is an alcohol.
Okay.
If you replace it with a carboxyl group instead, you get acetic acid, which is vinegar. That slight functional group substitution creates a completely different chemical identity and property.
Biological molecules are highly organized, which leads us to classify them based on their function and metabolism. The text breaks it down into 4 categories.
Right. First, you have the macromolecules, the polymers. These are the huge organized structures that carry out life proteins, nucleic acids, polysaccharides, and some lipids. The first 3 are built through polymerization, which the book likens to coupling railroad cars in Figure 2.11a. Monomers are just linked together repeatedly.
And second, the building blocks themselves, the monomers.
These are the low molecular weight precursors. We're talking sugars, amino acids, nucleotides, and fatty acids. These are what are continually synthesized and then recycled by the cell to assemble the macromolecules.
Third, we have the metabolic intermediates, or metabolites.
These are compounds that are formed along the step-by-step pathways of metabolism. They're very transient, they exist only as steps toward a final functional product, and they generally have no intrinsic function of their own.
And finally, a small catch-all group of miscellaneous molecules.
Yeah, this includes regulatory molecules like hormones and essential high-energy compounds like ATP and vitamins, which often function as cofactors for proteins. But it's really the macromolecules and their direct precursors that make up the vast bulk of the cell's dry weight. And the book also notes in Figure 2.11b that they are disassembled by the reverse process, hydrolysis.
Before we start building the final structures of life, we need to ask where the essential non-carbon elements come from. We all know plants perform photosynthesis, fixing carbon from TCO2O2, which the text says we'll cover in Chapter 6, but they critically need nitrogen and phosphorus to build proteins and DNA.
And this is a major chemical challenge for plant life. Nitrogen is particularly difficult because even though the atmosphere is 78% nitrogen gas, plants can't fix it themselves. They are completely reliant on specialized soil microorganisms.
Such as the bacteria that live symbiotically in the root nodules of legumes like peas and beans.
Correct. Rhizobia bacteria, living as what are called bacteroids inside these root nodules, perform the complex chemistry of converting atmospheric nitrogen to ammonia, which the plant can then utilize.
And this is why crop rotation, planting legumes to enrich the soil, has been a cornerstone of agriculture for millennia.
It is. Nitrate is also released when dead plant material decomposes in the soil.
But modern agriculture often relies on industrial intervention fertilizers.
Yes, either in the form of manure or, more commonly, synthesized ammonium nitrate. This industrial synthesis requires reacting natural gas with atmospheric nitrogen at very high temperatures and pressures. It's a massive industrial effort just to solve a biological problem.
Phosphorus seems to be an even trickier chemical problem because it doesn't come from the air at all.
Right. Phosphorus is absolutely essential for ATP, DNA, and RNA. It originates from rocks and minerals, specifically a compound called hydroxyapatite, which slowly leaches into the soil and groundwater.
And it gets used up.
It does. Because plants consume it and it's carried away by runoff, and there is no mechanism for air fixation, phosphorus rapidly becomes depleted from agricultural soil.
So phosphorus fertilizers have to rely entirely on geological sources.
They do. Sources like bone meal or mineral rocks that have been treated with acid to release water-soluble phosphate, and the source highlights the geopolitical significance here. The main global reserves of these phosphorus minerals are concentrated in just a few locations, primarily Morocco, China, and the U.S. This makes this one resource a major global economic and agricultural bottleneck. Hashtag 2.7, 4 types of biological molecules.
All right, now we launch into the main event. The grand tour of the 4 primary classes of biological macromolecules, starting with the fuels and the durable materials. Carbohydrates, hashtag A. Carbohydrates, glycans.
Carbohydrates, or glycans, are the primary energy stores and structural building materials for life. Their basic chemical formula is pretty simple, and we categorize them by the number of carbons, so you have trioses, pentoses, hexoses, and so on.
The key chemical characteristic is the large number of hydroxyl groups that are attached to the carbon backbone.
Yes, and these hydroxyl groups are what make sugars highly polar and extremely water-soluble. Sugars also feature a carbonyl group. If this group is internal, the sugar is a ketose, like fructose, which is shown in figure 2.14b. If it's at the end of the chain, it's an aldose, like glucose, shown in 2.14a.
And most sugars with 5 or more carbons don't actually exist as a linear chain, do they?
No, in solution, they spontaneously form closed ring-containing molecules. You can see this for glucose in figure 2.14c and d. The vast majority of sugar molecules are in this ring form. However, that tiny fraction that remains in the linear form is actually medically important.
How so?
Well, high glucose levels in diabetic patients allow that linear form of glucose to react non-enzymatically with hemoglobin. This forms a modified protein called TEX-HbA, which is used clinically to track long-term blood sugar control.
And we run into chirality again here, the mirror image problem.
We do. When a carbon atom bonds to 4 different groups, it becomes asymmetric, which creates enantiomers, or mirror images, as we see in figure 2.15. Life almost exclusively uses the D sugars, as shown in figure 2.16. And when glucose forms a ring, the C1 carbon becomes a new center of asymmetry, generating 2 stereoisomers called alpha and beta, which figure 2.17 illustrates. This tiny chemical difference is absolutely critical to biology.
And these sugars link up via covalent glycosidic bonds.
Right. Linking 2 monomers creates a disaccharide. Sucrose, or table sugar, which is shown in figure 2.18, is a disaccharide featuring an alpha linkage. Lactose, or milk sugar, has a beta linkage,
And to break that beta linkage in lactose, you need the enzyme lactase.
which many adult humans lose the ability to produce, and that leads to lactose intolerance. Oligosaccharides, which are short chains of sugars attached to proteins, or lipids, glycoproteins, or glycolipids, are crucial informational molecules on the cell surface. They act like identity markers.
And linking many monomers together gives us polysaccharides, which can be nutritional stores or structural elements.
Exactly. For nutritional stores, we can look back to Claude Bernard, the 19th century physiologist who discovered glycogen.
Glycogen is the animal storage form.
Right. It's a highly branched glucose polymer used for energy storage in animals, primarily in the liver and muscles. You can see its branch structure in figure 2019a. It features both alpha-1 to 4 and alpha-1 to 6 linkages. Plants store glucose as starch, which figure 2.1b shows is a mix of unbranched amylose and branched amylopectin. Both are easily digestible by humans.
But structurally, we find molecules that are essentially identical in composition, yet they're completely indigestible to us.
Cellulose is the perfect example. It's also a polymer of glucose, just like starch. But instead of the alpha 1 to 4 bond, it uses the beta 1 to 4 linkage, which you can see in figure 2.19c.
And that small change makes all the difference.
All the difference. It results in tough, unbranched fibers, the main component of plant cell walls, and what we call dietary fiber, that are resistant to almost all degradation. Only organisms with the specific enzyme cellulase, like the symbiotic microbes in the guts of cattle and termites, can break it down.
What other structural polysaccharides do we rely on?
Well, there's chitin, which forms the hard but flexible exoskeleton of insects and crustaceans. As figure 2.2z shows, it's an unbranched polymer of a modified sugar, N-acetylglucosamine. And then we have glycosaminoglycans, or GAGs, which are repeating disaccharide polymers. Heparin is a famous GAG that's used clinically as an anticoagulant because of its ability to inhibit blood clotting. #B lipids.
Next up, the lipids. They're defined not by a common structure, but by their shared nonpolar characteristic. They are soluble in organic solvents, but insoluble in water. They are the dense energy stores and the architects of the cell membrane.
Lipids include fats, steroids, and phospholipids. Fats, or triacylglycerols, are built from a glycerol backbone that's linked by 3 ester bonds to 3 fatty acid chains. You can see this in figure 2.21a.
And fatty acids are amphipathic, meaning they have split personalities.
They do. They have a long, nonpolar hydrophobic hydrocarbon tail and then a polar hydrophilic carboxyl head.
And this is the chemistry that makes soap work.
It is. Soap consists of fatty acids that form micelles in water, which figure 2.22 illustrates beautifully. They trap nonpolar grease and dirt in their core while presenting their polar heads to the water, which allows them to dissolve the grime.
And the saturation of those fatty acid tails affects their physical state.
It does. Fatty acids without any double bonds are called saturated, as in figure 2.21c. They can pack tightly together, which results in solid fats like butter or lard at room temperature.
Whereas unsaturated fats have kinks.
Right. Unsaturated fatty acids contain double bonds, which introduce kinks in the chains, as you can see in 2.21 feet. This prevents tight packing and results in liquids like vegetable oils. The industrial process of hydrogenation, which is used to make vegetable shortenings, converts some of those natural double bonds into trans double bonds, creating trans fats that are now linked to increased cardiovascular risk.
And fats are superior energy storage molecules.
They are. They contain over twice the energy per gram compared to carbohydrates. And because they're nonpolar, they are stored dry, without water, in these dense, concentrated lipid droplets. That makes them the body's ideal solution for long-term energy storage.
We also have steroids, which are built around a really unique architectural skeleton.
Steroids are defined by their characteristic four-ringed hydrocarbon structure. Figure 2.23 shows cholesterol, which is the most important example. It's vital for maintaining the fluidity and structure of animal cell membranes. It also serves as the necessary precursor for synthesizing all the steroid hormones, like cortisol, testosterone, and estrogen.
And finally, the phospholipids, the structural heart of the membrane.
Phospholipids are structurally similar to fats, but they're diacylglycerols, meaning they only have two fatty acid chains. The third position on the glycerol is instead linked to a phosphate group, which then connects to a small polar head group like choline. Figure 2.24 shows this structure.
And this makes the phospholipid strongly amphipathic.
Very strongly. It has a distinct hydrophilic head and two hydrophobic tails. And this structure is the fundamental property that causes them to spontaneously assemble into the lipid bilayer that forms every single cell membrane. #C, building blocks of proteins.
We come now to proteins, the most functionally diverse molecules. They act as the tools, the machines, and the catalysts for virtually every single cellular process, and their activity is defined entirely by their specific, unique shape.
That's right. Proteins are polymers built from 20 different L-amino acid monomers. Each amino acid has a common backbone, an amino group, text NH3 plus 9, and a carboxyl group connected to a central alpha carbon. You can see this in figure 2.26. They are joined by peptide bonds to form a continuous polypeptide chain that runs from the free amino group, the N-terminus, to the free carboxyl group, the C-terminus.
And the incredible diversity comes entirely from the unique side chains, or R groups, that are attached to that alpha carbon.
The R groups are everything. We categorize them based on their chemical nature, and figure 2.28 lays them all out.
Can you walk us through the categories?
Sure. First, you have the polar charged side chains. These are highly reactive organic acids or bases like lysine or glutamic acid. They're usually fully charged at physiological pH and participate in ionic bonds. You often find them in enzyme active sites, as figure 2.29 shows.
Okay.
Second, the polar, uncharged. These, like serine or threonine, have partial charges and they can form crucial hydrogen bonds. Third, you have the nonpolar side chains, like alanine and valine. These are hydrophobic. They cluster together in the protein's core, stabilized by van der Waals forces, and this clustering is the fundamental driving force for protein folding.
And there's a fourth, unique category.
Right. This includes glycine, which is the smallest and offers a lot of flexibility, proline, which forms a ring that introduces kinks into the chain, and cysteine, which can form stabilizing covalent disulfide bridges with other cysteine residues.
Disulfide bridges are covalent, which makes them much stronger than the noncovalent bonds. They're often used for stability in proteins that have to function in harsh environments outside the cell.
Exactly. They lock the structure in place. For instance, the toughness of hair keratin is due to its high concentration of these cysteine crosslinks. When you get a perm, the stylist is chemically breaking and then reforming those disulfide bridges to lock your hair into its new shape.
And once it's synthesized, a protein is often modified via what are called post-translational modifications, or PTMs.
Yes, PTMs are reversible alterations to the side chains that happen after the polypeptide is made, and they act as powerful regulatory switches. The most common one is phosphorylation, the reversible addition of a phosphate group, usually to serine, threonine, or tyrosine residues.
And one small change can have a huge effect.
A huge effect. This single chemical change can instantly and dramatically alter a protein's 3D structure, its activity, or even its localization within the cell.
So when a protein folds up in water, the location of these different side chains is highly predictable based on their chemistry, isn't it?
It is. As figure 2.30 illustrates, the hydrophilic, so the polar and charged residues, seek out the surface of the molecule to interact with the surrounding water. This is what ensures the protein's solubility.
And the hydrophobic ones hide inside.
Exactly. The hydrophobic nonpolar residues are buried deep in the core. This internal cluster, but it also creates a unique chemical microenvironment within the core, which can often enhance the catalytic power of the enzyme's active site. Hashtag primary and secondary structures.
Okay, so the first level of organization in a protein is its primary structure, the linear sequence of amino acids.
And this sequence dictates everything that follows. The potential diversity is nearly infinite, and the importance of even a single one of these residues is dramatically illustrated by sickle cell anemia.
Right, sickle cell anemia is caused by a single amino acid substitution in the hemoglobin protein.
A single genetic mistake replaces a charged glutamic acid residue with a nonpolar valine residue. You can see this in figure 2.31. This tiny change in the primary sequence causes a massive conformational disruption in the hemoglobin molecule, which in turn causes red blood cells to deform into that characteristic sickle shape.
Leading to all the symptoms of the disease.
Yes, vascular occlusion and the acute crises associated with the disease, all from one amino acid.
So moving up to the secondary structure, which describes the conformation of portions of that chain, we look at the regular repeating structures discovered by Linus Pauling and Robert Corey.
Right. They realized that the backbone of the polypeptide naturally favors conformations that maximize the hydrogen bonds that can form between the peptide bonds themselves. The two most common structures are the alpha helix and the beta sheet.
The alpha helix is that twisting spiral.
It is. As you can see in figure 2.32, it's stabilized by hydrogen bonds that run roughly parallel to the helix axis. They link the carbonyl group of one peptide bond to the imine group of the peptide bond that's 4 residues further along the chain. This forms a stable cylinder with all the R group side chains projecting outward.
And the beta sheet is the pleated structure.
Yes, this consists of multiple segments, called beta strands, that are lying side by side. Here, as figure 2.33 shows, the hydrogen bonds project perpendicularly across the chains. These sheets are incredibly stable and strong. The remarkable toughness of spider silk, for example, is attributed to its highly organized beta diarsheet structure. Hashtag tertiary structure of proteins.
The tertiary structure is the overall 3D conformation of the entire polypeptide chain, and it's stabilized by all those non-covalent bonds between the side chains. How do we even determine these complex shapes?
Well, for decades, the standard method was X-ray crystallography. This requires growing a pure protein crystal and then analyzing the diffraction patterns that are created when X-rays bombard it. That pattern is then used to mathematically deduce the atomic coordinates. You can see an example of the setup in figure 2.35.
There's also a solution-based alternative, nuclear magnetic resonance, or NMR spectroscopy.
Right. NMR probes proteins in solution and reveals the distances between atoms. As figure 2.36 shows, NMR is great for seeing dynamic changes in a protein, but it really struggles with very large proteins.
And now, of course, there's the game-changing technology of cryo-electron microscopy, or cryo-EM.
Cryo-EM has absolutely revolutionized the field. By embedding proteins in a layer of non-crystalline ice and taking millions of low-dose images, computers can generate atomic resolution structures without ever needing to grow a crystal. This is vital for studying large, flexible, or dynamic molecular machines that just won't crystallize.
And it's also been discovered that not every single part of a protein actually has a fixed structure.
Exactly. Many proteins contain what are called intrinsically disordered segments. These are flexible, almost spaghetti-like regions that lack a defined conformation until they bind to a specific partner molecule. Upon binding, they immediately fold into a fixed structure, and this shows their crucial role in dynamic processes like signaling pathways.
The very first detailed tertiary structure ever solved was myoglobin by John Kendrew in 1957.
Yes, Kendrew showed that myoglobin, which stores oxygen in muscle, was a compact molecule, mostly alpha helical. Figure 2.37 and 2.38 show its structure, and it was held together entirely by non-covalent interactions. This was the first proof that each protein adopts a single, unique, predictable tertiary structure that's directly related to its function.
Large eukaryotic proteins are often built from distinct functional modules called domains.
That's right. Domains are modules that can fold independently of the rest of the chain. For example, the enzyme phospholipase C, shown in figure 2.40, has 4 distinct domains. Each one likely carries out a semi-independent function or a binding activity.
And this is important for evolution.
Very. Evolution frequently creates new proteins by a process called domain shuffling, fusing the genes that encode different ancestral domains together to rapidly generate new complex functions.
The solved structures often present a static picture, but proteins are flexible, dynamic machines. What's actually happening inside them?
Well, they are constantly undergoing thermal motion. We can use molecular dynamics or MD simulations to model this movement computationally. Take the enzyme acetylcholinesterase, for instance. MD simulations, like the one depicted in figure 2.41, showed that its catalytic site is buried deep within the molecule.
So how does the substrate get in?
Thermal fluctuations cause temporary gates to open and close, allowing the substrate to diffuse in and the product to diffuse out.
And these non-random movements that are triggered by binding are called conformational changes.
Yes, and conformational changes are crucial. When the bacterial chaperonin GroEL binds its cap protein, GroES, it undergoes a dramatic, predictable conformational change. Similarly, muscle contraction relies on millions of tiny, predictable conformational shifts in proteins like myosin and actin. Hashtag quaternary structure of proteins.
Quaternary structure is the highest level of organization. Proteins that are composed of multiple polypeptide chains or subunits.
These subunits can be identical, which we call homodimers, or non-identical, which would be heterodimers or heterotetramers. They're held together primarily by complementary surface interactions and noncovalent bonds. The classic example here is hemoglobin.
Right. Hemoglobin, which carries oxygen in the blood, is a heterotetramer. It's made of 2 alpha and 2 beta globin chains.
And Max Peretz's work, visualized in figure 2.42b, showed that oxygen binding causes a huge coordinated conformational shift in the entire tetramer. Then the movement of the iron atom in the heme group pulls on an alpha helix, which triggers a shift that alters the affinity of the other 3 subunits for oxygen. This dynamic interaction is called cooperativity.
Beyond individual functional proteins, cells assemble these massive complexes, molecular machines that can channel substrates efficiently.
Yes, the bacterial pyruvate dehydrogenase complex, for instance, shown in figure 2.43, consists of 60 polypeptide chains making up 3 distinct enzymes.
And why is that useful?
Because these enzymes are physically associated, the product of the first enzyme is passed directly to the active site of the second. This prevents the product from being diluted in the cell's aqueous environment and makes the whole process much more efficient.
And these interactions between partner proteins require a precise molecular fit, which is often mediated by what are called adapter domains.
Adapter domains, like the SH3 domain shown in figure 2.44, feature these shallow hydrophobic pockets that specifically recognize and bind to complementary shapes on their partner proteins. And these highly specific interactions are often regulated by PTMs like phosphorylation, which can act as a molecular switch to turn the binding capability on or off. Hashtag protein folding.
So how does a protein arrive at its specific functional 3D structure? The information for that fold has to be encoded somewhere.
It is. The information is contained entirely within the linear primary sequence. This was proven decisively by Christian Anfinson's 1956 experiment with the enzyme ribonuclease A.
What did he do?
He completely unfolded the protein, he denatured it, using urea and mercaptoethanol, which caused it to lose all of its activity. The process is diagrammed in figure 2.45.
And then, upon removing the denaturants, it spontaneously refolded.
It did. The protein regained its native active state all by itself. This proved that the amino acid sequence contains all the necessary instructions for self-assembly. The protein simply adopts the most thermodynamically stable conformation, the one with the lowest possible energy.
But the actual dynamics of folding are much faster and more complicated than this simple picture suggests. The book mentions a couple of models in figure 2.46.
Right. The current view of folding involves a simultaneous compaction and formation of secondary structures, which leads to what's called a transition state structure depicted in figure 2.47. This structure is already very similar to the native protein, but it just lacks the final precise packing.
And this happens incredibly fast.
On a microsecond timescale, it shows how incredibly efficient life is at maximizing these non-covalent interactions to find that lowest energy structure.
Now, if the sequence dictates the fold, then any mistake in that process can be catastrophic. And this leads us to a range of fatal neurodegenerative diseases, including prion diseases and Alzheimer's disease.
Yes, the chapter's second human perspective box covers this in detail. Prion diseases like Creutzfeldt-Jakob disease or CJD are unique because they can be inherited, sporadic, or even acquired like in mad cow disease. The infectious agent is the prion, which is the focus of the protein-only hypothesis.
So the normal prion protein, TEX-PPP, is soluble and rich in alpha helices, but the infectious version is misfolded and rich in beta sheets.
And critically, the two molecules can have the exact same amino acid sequence. But text is folded into a stable, insoluble, aggregate-forming conformation. The template conversion model, which is shown in Figure 1 of the box, suggests that when an infectious text encounters a normal text, it forces the normal protein to refold into that infectious beta sheet-rich structure.
Starting a chain reaction.
A chain reaction that kills nerve cells, leading to what's called spongiform encephalopathy.
Alzheimer's disease is far more common, and it also involves these fibrillar deposits of insoluble material called amyloid.
Right. Alzheimer's involves the amyloid beta peptide, or A-beta. This peptide is cleaved out of a larger protein called ATT by two specific enzymes, which you can see in Figure 3 of the box. The critical species seems to be a beta 42, which has two extra hydrophobic residues, and it spontaneously refolds into a beta sheet-rich structure.
And this beta 42 is toxic.
Yes, it aggregates into toxic soluble oligomers and also forms the extracellular amyloid plaques that are the hallmark of the disease, shown in Figure 2 of the box.
The fact that all the known genetic mutations that lead to early onset Alzheimer's increase the production of this toxic beta 42 peptide is the strongest support for the amyloid hypothesis. But the translation of this theory to successful drugs has, well, it's failed dramatically.
It's been incredibly difficult. Strategies that were aimed at removing the existing A-beta aggregates using either vaccines or passive antibodies like bapineuzumab and solanezumab have repeatedly failed in phase 3 clinical trials.
What did the analysis of those patients show?
It showed that while their plaques were successfully cleared from the brain, their cognitive decline did not halt.
So that suggests either that the plaques are not the primary cause of the symptoms, or that by the time symptoms appear, irreversible damage has already occurred. This has forced a major shift in focus to the other misfolded protein in Alzheimer's brains, tau.
Yes. Tau forms tangled filaments called neurofibrillary tangles, or NFTs, inside the nerve cells. While the A-beta mutations clearly trigger the disease, the burden of NFTs actually correlates much better with the extent of cognitive decline and neuronal loss than the plaques do. Reconciling the A-beta cause with the tau correlation remains a major research challenge.
So if Anfinsen proved that folding is spontaneous, why does a cell need helper proteins or molecular chaperones to assist in the process?
It's a great question, and the focus of the experimental pathways box. The reason is that the cell is an incredibly crowded place. An unfolded protein is very vulnerable to non-selective, sticky interactions with other macromolecules, particularly via its exposed hydrophobic residues.
Which leads to aggregation.
Immediate aggregation and deadly misfolding. Chaperones selectively bind to these exposed hydrophobic patches, preventing that aggregation and allowing the protein sufficient time and space to find its correct conformation.
And the discovery of these chaperones emerged from studies on the heat shock response.
Yes. FM Ratosa found that increasing the temperature in fruit fly larvae activated genes that produce what he called heat shock proteins, or HSP. And then independently, researchers studying bacterial virus assembly found that the phages required host proteins called GroE, which were later identified as the chaperone system GroEL and GroES, to assemble correctly.
The two major chaperone families are Hsp70 and Hsp60, and Hsp70 seems to catch the nascent chains right as they're being made.
That's right. Hsp70 proteins bind to emerging polypeptide chains as they exit the ribosome, as you can see in Figure 2.48. They shield the hydrophobic patches and prevent premature aggregation. Smaller proteins might fold spontaneously upon release, but larger, more complex proteins are often handed off to the cylindrical Hsp60 chaperonins, such as GroEL.
And GroEL is often described as this cylindrical double donut with a central chamber, and GroES acts as the cap.
Yes, the complex is composed of 14 subunits that form two rings, with an interior cavity large enough to encapsulate a folding protein. You can see the structure in Figure 1 of the box. When the cap GroES binds, as shown in Figure 2, it triggers a dramatic 60-degree rotation of the apical domains of the GroEL subunits.
And that structural change is the key to the whole mechanism.
It is. The conformational change, which you can see in Figure 3, dramatically enlarges the internal chamber, and crucially, it changes the chamber's internal surface chemistry from hydrophobic, which is what binds the non-native protein, to polar.
Which releases the protein.
It releases the non-native protein into this protected, enlarged space, giving it the opportunity to fold correctly without any external interference. The whole cycle is detailed in Figure 4.
And there's also evidence that the chaperonin plays an active rather than just a passive role in folding.
There is. Studies using a technique called FRET, which measures distances between fluorescent tags on a protein, showed that when GroEs binds, the GroEl cavity can actually forcibly unfold a substrate protein.
So it's like hitting a reset button.
Exactly. This resetting ensures the protein is taken all the way back to an unfolded state and given a complete restart, preventing it from getting trapped in a misfolded intermediate. But this powerfully reinforces Anfinson's initial finding. Chaperones prevent disaster, but the ultimate 3D structure is still dictated by the sequence itself. Hashtag proteomics and interactomics.
Inventorying all the proteins produced by an organism, its proteome, has led to the field of proteomics. And this is much, much harder than simply reading the genome.
It is. Proteins are incredibly chemically diverse, they're difficult to amplify, and they're present in wildly varying concentrations. The key technology to identify them is mass spectrometry, or MS.
So how does MS identify an unknown protein?
The protein is first broken down into smaller fragments, or peptides, using an enzyme like trypsin. The MS machine then measures the precise mass-to-charge ratio of these fragments, which generates a highly characteristic peptide mass fingerprint. You can see an example of this in figure 2.49.
And then you match the fingerprint.
Right. Researchers compare this unique fingerprint against a computerized database of all the theoretical proteins encoded by the organism's genome, and that allows them to identify the protein with great accuracy.
Proteomics has clear application in medicine, particularly in biomarker discovery.
Yes, by comparing the proteome of a diseased sample with a healthy one, we can discover protein patterns or biomarkers that indicate the presence of a disease. For instance, mass spectrometry data helped lead to the development of tests for ovarian cancer, providing critical information before a patient even has surgery.
Beyond just an inventory, researchers also want to map the complete network of all the protein-protein interactions, the interactome.
A standard method for mapping the interactome is called TAP-P-TAG mass spectrometry. Here, a protein of interest is chemically tagged, it's purified from the cell, and then any partner proteins that co-purify with it are identified using MS.
And the results are visualized as these complex network diagrams.
Exactly, like the one in figure 2.5. And these networks reveal that certain proteins act as hubs interacting with many, many partners.
And these hubs are usually essential proteins.
Often, yes. And interestingly, the complexity of these hubs can vary. Some, like RNA polymerase II, have multiple distinct binding interfaces and can bind many partners simultaneously. Others, like the cell cycle regulator CDC28, which is shown in figure 2.51, have a single binding interface and have to switch between partners one at a time. Hashtag protein engineering and adaptation.
Our incredible knowledge of protein structure now allows us to move beyond just studying existing proteins to actively designing new ones. Protein engineering.
We can synthesize artificial genes for any sequence we choose. The real challenge is computing which sequence will actually fold into a stable, useful 3D shape. But computational simulations now allow scientists to design proteins that can bind specifically and with high affinity to target molecules.
Like what?
For example, the text shows a protein in figure 2.52 that was designed to inhibit the hemagglutinin protein of the influenza virus.
But the most profound advancement is the creation of de novo enzymes, enzymes that are capable of catalyzing reactions that no natural enzyme has ever performed.
This is truly amazing. Researchers use computational techniques to design the geometry of an active site and then search through known protein structures for a scaffold that can hold that site. The artificial proteins that have been created can accelerate specific organic reactions. This proves that we can now, in theory, construct proteins capable of catalyzing virtually any desired chemical reaction from scratch.
And this structural knowledge is also the basis for what we call structure-based drug design.
Yes. By knowing the precise tertiary structure of a disease target protein, scientists can computationally design virtual molecules that will fit tightly into its active site and inhibit it. The prime example of this is Gleevec, or imatinib, which is shown in figure 2.53.
A drug that revolutionized the treatment of chronic myelogenous leukemia, CML.
It did. Gleevec inhibits the ABL tyrosine kinase by locking it into an inactive conformation. And this whole process relies on initially identifying a weak compound and then computationally improving its fit and affinity based on the known structure of the target.
Finally, looking at evolution, proteins have to adapt their structures to extreme environments.
They do. While homologous proteins diverge very slowly in their 3D structure, their primary sequence can change quite rapidly. For example, proteins in halophilic or salt-loving archaebacteria have highly acidic surfaces, as you can see in figure 2.54. They're coated with charged glutamic and aspartic acid residues, and this is what maintains their solubility in the extremely high cytosolic salt concentrations they live in.
And even a single amino acid change can have huge evolutionary consequences.
It can. The text notes with figure 2.55 that it's possible for a single amino acid substitution to cause a massive transformation in the entire fold of a small domain, potentially generating the ancestral form of an entirely new protein family. #D nucleic acids.
The fourth and final class of macromolecules,
Mhm.
nucleic acids, the molecules dedicated to information storage and transmission.
Nucleic acids are polymers that are built from nucleotide monomers. And a nucleotide consists of three parts, which are laid out in figure 2.56a, a 5-carbon sugar, ribose and RNA, deoxyribose and DNA, a nitrogenous base, and a phosphate group.
And the bases are either 2-ring purines, adenine, guanine, or 1-ring pyrimidines, cytosine, uracil in RNA, or thymine in DNA. The structures are all in figure 2.57.
Right, and these monomers are linked together via 3R, 5R phosphodiester bonds, which you can see in figure 2.56b. These form the robust sugar-phosphate backbone of the chain.
We often think of RNA as just a simple single strand, but functionally it is far more complex than that.
It is. RNA may be synthesized as a single strand, but it folds back on itself expensively, forming these complex 3D structures with localized double-stranded regions that are stabilized by base pairing. You can see an example of this in figure 2.58.
And these highly structured RNA molecules can even possess catalytic activity. They're called ribosomes.
Yes, the hammerhead ribozyme, for instance, is an RNA molecule that's capable of self-cleavage. This discovery completely challenged the historical assumption that all cellular enzymes had to be proteins.
And nucleotides also have important functions independently of being polymers.
They do. They're critical energy molecules. ATP, adenosine triphosphate, is the universal energy currency for the cell, and GTP, guanosine triphosphate, functions as a molecular switch, binding to G proteins to turn their activities on and off in various signaling pathways. #tag 2.8, the formation of complex macromolecular structures.
We've covered how individual proteins fold, but how do huge complexes like viruses or ribosomes that are made of hundreds of different components, how do they assemble themselves correctly?
Well, the simplest model is self-assembly, which means that all the necessary information for assembly is contained within the component subunits themselves. A famous early proof of this came in 1955 when researchers showed that tobacco mosaic virus, or TMV components,
Just RNA and protein.
right, it's RNA and 2130 identical protein subunits could spontaneously form infective viral particles in vitro just by mixing them together in a test tube.
And bacterial ribosomes also demonstrate a high capacity for this kind of self-assembly.
They do. Nomura showed that the bacterial 30S ribosomal subunit could be fully reconstituted in vitro from its constituent rRNA and 21 different proteins.
So the information is all there.
The information is present in the components, but it's important to note that the assembly still requires accessory factors in a living cell just to speed up the process. Eukaryotic ribosomes, which are much more complex, require extensive processing and they cannot self-assemble in vitro.
And finally, a relatively new insight into cellular organization, these things called phase-separated compartments. These are non-membrane-bound organelles.
Yes, this involves a process called aqueous phase separation. This is where soluble polymers, proteins, and RNA separate into distinct liquid-like droplets within the cell, kind of like oil droplets in water. This is increasingly being recognized in structures like the nucleoli, which is the site of ribosome assembly, and compartments that contain RNA splicing factors like the FUS protein.
So how do these liquid droplets form and why are they necessary? What's the point?
Their formation seems to be driven primarily by proteins that contain those intrinsically disordered domains we talked about, and also weak transient interactions between RNA and protein. The key biological consequence is that this phase separation creates extremely high local concentrations of molecules within the droplet.
So it brings everything together.
It brings everything together, promoting molecular interactions far more effectively than if those same molecules were diluted across the entire volume of the cell. It's a way to spatially organize a chemical reaction without needing a membrane.
And these dynamic droplets can even predict future cellular changes.
They can. The location of certain droplets, such as those formed by the We3 protein, which you can see in figure 2.60, has been shown to predict future cellular events, like where a new branch will emerge on a fungal filament.
That's fascinating.
It is. Now, when these weak interactions are stronger, the compartment can transition from a liquid-like state to a more elastic gel, a process called gelation, which provides more structural integrity. Proving the absolute biological necessity of these liquid compartments is currently a very highly active area of research. Hashtag Hag Outro. So to synthesize this entire foundational deep dive, we've established that life is founded on this precise interplay between the strong, stable covalent bonds, which form the backbone of molecules, and the dynamic, weak, non-covalent interactions, ionic, hydrogen, hydrophobic, and van der Waals, that enable movement, interaction, and assembly.
And this entire system operates within the critical matrix that's provided by water and regulated by buffers.
Exactly.
We mapped the architectural diversity of the four macromolecules: carbohydrates for energy and structure, lipids for membranes and energy storage, nucleic acids for information, and of course proteins, the molecular machines whose primary sequence dictates their complex functional 3D structure. And crucially, we saw that while that folding is spontaneous, the crowded cellular environment demands the assistance of chaperones to prevent the destructive misfolding that we see in so many neurodegenerative diseases.
Right. The primary sequence holds the key to the functional tertiary structure, but it's the cellular environment that makes that function possible.
We've seen that scientists can now use this knowledge to design entirely new proteins and enzymes from scratch, potentially accelerating chemical reactions that life has never even encountered. So, given that researchers can utilize test tube evolution to improve these artificial proteins, how might the rapid design and evolution of novel biological catalysts impact our understanding of natural selection? And what are the long-term implications of introducing functions that are completely outside of life's established design constraints?
A provocative thought for you to explore further.
For now, that's all the time we have for this deep dive into the chemical basis of life, and a warm thank you from the Last Minute Lecture team.